Welcome. Thanks for joining. My name is Avi Srivastava, and today I'm going to talk about a Bayesian algorithm for droplet-based single-cell RNA-seq quantification. This work was done as part of my PhD thesis at Stony Brook University under Rob Petro. In a typical droplet-based single-cell experiment, you've been provided with paired FASTQ files. In the paired FASTQ files, one end represents cellular barcode and the UMI. The second end represents transcript sequences. The problem of quantification involves grouping the set of reads, first based on the cellular barcodes, then based on the UMIs to generate gene by cell count matrices. In theory, this can be done relatively easily using a couple of lines of pandas or Python code. But in practice, this can be much more complicated. The complication can occur due to multiple reasons. For example, what if there is a PCR error or sequencing error? Or third, most importantly, what if there is a multi-mapping read? That is, how do you assign a read which maps equally well to more than one gene? A typical solution to this problem is drop them away. However, last year we published Elvin, which modeled these multi-mapping reads in a principal framework and provide an end-to-end pipeline which starts with raw FASTQ files and generates cell versus gene count matrices. One obvious question comes up: how frequent are these gene multi-mapping reads? And that's what we show on the left table. The number of gene multi-mapping reads can be as high as 23% of the data, which is a quarter of your data set. And tools which drops them away are consistently creating a bias toward a subgroup of genes which have highly ambiguous gene sequences, which is a problem. And that's what we model in our Elvin framework. We perform cellular barcode sequence correction, we perform transcriptome mapping, we perform UMI deduplication, and generate cell versus gene count matrix. One important point to mention here is the UMI deduplication step involves parsimony-based condition where we resolve pre-PCR molecules into a group to calculate the gene versus cell count matrix. The parsimony-based condition of Elvin adjudicates multi-mapping problem in a direct way by finding a simplest solution to the problem. However, it's still possible to have multi-mapping reads even after the resolution. And we solve it by using expectation maximization algorithm. Just to give a brief summary, Elvin divides the problems into 3 tiers based on their difficulty. Here, I'm showing at the top 2 transcripts, T1 and T2, coming from 2 genes, G1 and G2. The first version of the problem, or the easiest version of the problem, is when a gene G1 has all unique mapping reads and the second gene G2 also have all unique mapping reads. The problem is easy and both of them can have separate counts. A difficult version of the problem is when some subgroup of the genes have unique and some subgroup of genes have multi-mapping reads. Here I'm showing gene 2 has 2 unique reads and there is 3 reads which maps equally well to gene G1 and G2. So how do you assign these reads to which of one of the reads? It's a complicated problem. The intuitive understanding would be to assign more reads to a gene which have a unique evidence and downweigh the assignment to the gene which has no unique evidence. And these are kind of the problem which our expectation maximization algorithm can solve reasonably well. But there is a third category of problem, Elvin called them tier 3 category problem, when there is no unique evidence for any of the genes. The tier 2 category problems can be solved reasonably well using expectation maximization algorithm. In this, we have been given a set of known genes G, a set of gene level equivalence classes E, where equivalence classes represent a subgroup of genes where a read maps and cannot disambiguate between them. Also, we have a gene UMI binary assignment matrix along with a set of deduplicated UMI's calligraphic U. What we have to find is the probability of generating a molecule from each gene. Here, I'm denoting it by rho. What basically we have to find is the probability of a row given U and G. We can always use Bayes' rule, which will make it directly proportional to probability of observing a UMI given gene. And we also have a prior distribution term. Here we are going to use uniform distribution. How we define the likelihood depends on probability of observing U given G, and it's a multiplicative probability for E gene GI given the expression pattern. Intuitively, it comes down to the similar tier 2 example which we saw before. We use the unique evidence as a weighing factor to distribute genes among the equivalence classes which have multi-mapping. This was great. However, the tier 3 category problems are still there. And we start thinking how we can model them more efficiently. And we came up with this hypothetical experiment where we want to look at two cells where Oracle tells us they are coming from the same cell type. It is important because in expectation, we expect the gene expression pattern across the same cell type would be the same. Within the experiment, I wanted to look specifically for gene A in both the cells. Since they're coming from the same gene, we expect them to have the similar exons, and that too sequentially. Before sequencing, there is a step of fragmentation which divides the molecule into small fragments. And since it happens uniformly at random, the molecules get chopped at random location. Assuming the top one got chopped at exon B and the bottom one got chopped at exon A, we will end up generating such kind of reads. Obviously, cellular barcode and the UMI is going to be the same. However, the read component can be different. The top one might have some exon B, which is shown by a violet color, while the bottom one will have only the gray color since it's coming from only exon A. Assuming if there was a reference with two genes, gene A and gene B, the gene A has both the exon A and exon B, while gene B only had exon A. Once we map read 2 of the top and the bottom cells, the top one map uniquely to gene A, while bottom will map ambiguously between gene A and gene B. And it will end up creating top one with tier 1 estimate since it's a unique mapping read, but with the bottom one with tier 3 estimate since we cannot resolve them among gene A and gene B. What if we can share the information? If we can share the information, then we can potentially improve tier 3 estimates. However, what if all the estimates are tier 3? It's not going to be useful. That's what I'm trying to show with this table. What categories would it be useful? Assuming the gene 3 situation where everything is tier 3, you cannot improve anything within a cell. Now, if one of them is T3 and everything is 0, then it's still not useful, although you can still use like this gene is not expressed. The interesting cases would be when a very small group of genes are tier 3, while everything other in other cells are tier 1. Then there is very good chance those can improve or transfer the confidence from tier 1 towards tier 3. And that's what we checked next. We looked in a real experiment and figured out how many tier 3 estimates have tier 3 as their neighbors. In the plot, we show that less than 10% of the tier 3 estimates has actually tier 3 and most of them are tier 1, which means we can use those tier 1 as a transfer from high confidence to low confidence cells. We have some intuition, we have some empirical evidence, then how are we going to model it? Going back to our expectation maximization algorithm, where we have a likelihood term and we were using uniform distribution as a prior. Now, what if we can alter this prior to transfer the cell from a high confidence cell to this one? For that, the first thing we would need, what are the nearest cells? And we use off-the-shelf algorithm here, Sura 3, to figure out what are the group of cells which are nearest to this cell of query. And if that has tier 1 estimate, we can transfer that knowledge to this as a prior distribution for improving the quantification estimates. Awesome. Now we have biological intuition some empirical evidence to work on, and also a good model to work with. How does it turn out in a real experiment? To do that, we have to first figure out what tools we should compare against. After Elvin, there are a bunch of tools which came out. For example, Star Solo, Hera T, and Bus tools. Before Elvin, there was Yumi tools and the Cell Ranger. A couple of important points to note here, that no other method except Elvin and Yumi tools has been published yet. Second, no other method is actually accounting for the gene multimapping reads. They are just dropping it away. And the third and the most important point, the tools which came out after Elvin, they show their validation by showing high correlation with Cell Ranger. It makes a task of validation a little easier, because if we can show Elvin is doing better than, if Elvin is doing better than Cell Ranger, then it's most probably doing better than other methods also. We went further and looked into PBMC 4K 10x dataset and simulated 4040 cells using Minnow. Minnow is an important tool which we published last year at ISMB, and it's specifically needed to validate our results in this case, because Elvin works on read level dataset, and if your simulator is working on gene counts level, it's not very useful. We designed Minnow to specifically account for PCR error and generate read level dataset with the ground truth, which can be used as a simulator. Now, in this dataset, we again use 4440 cells, simulate them using Minnow, and generate ground truth dataset. Later, we run Cell Ranger, Elvin with EM, and Elvin with VPEM. The important point is, since we know the ground truth, we can actually calculate the cell-wise Spearman correlation through the generated estimate by various methods. And that's what we did. And it's been shown in this plot, a cell-wise Spearman correlation of tier 3 estimates for EM, VBEM, and Cell Ranger, where VBEM is doing much better than EM, and specifically is doing much better than Cell Ranger. Great. We're doing much better in simulated dataset, but how about real dataset? It turns out non-availability of a standard or gold standard data makes the validation task in real dataset a bit harder. We need to come up with some ideas so that we can validate on the real dataset, and we came up with a knockout experiment. Imagine if you have a tier 2 kind of condition where you have some unique evidence and some ambiguous evidence based on gene 1 and gene 2, and I'm calling this as a full experiment. What if we knock out the unique evidence from the full experiment and generate a new experiment? We call it here as knockout experiment, where we only have the ambiguous evidence. If we quantify just based on the ambiguous evidence and still being able to produce quantification estimates closer to the full estimates, then our method is doing something better. Before we move on to results, I just want to show if a knockout experiment is even working. We take a real experiment and figure out what fraction of the estimates are tier 3 before and after performing a knockout. As you can see in this histogram, the ratio of cells with tier 3 estimates before knockout through blue histograms, it was much higher for the lesser fraction. But as we perform the knockout, the fractions of tier 3 genes increases dramatically. Okay, how do we validate our result on real dataset? What we did was we take PBMC 4K experiment and divide into two halves, equal halves, A and B, and run a knockout only on the first half or the A half. We can run Elvin regularly as on the knockout experiment, but to run VBEM, we need a prior distribution. We learn prior distribution by anchoring the knockout experiment on the second half of the experiment and quantifying just the first half using the prior learned from the second half to call it as a VBEM experiment. And we compared the knockout experiment quantification with the full experiment quantification to see how do they correlate. On the right, I'm showing the Spearman correlation of cell-wise Spearman correlation of the knockout experiment with the full experiment. And as expected, VBEM is doing much better than the EM algorithm. With this, I would like to conclude the presentation by mentioning some of the important points we discussed. In this presentation, we proposed a generic framework to share information across cell, and we show example how we can share information within one experiment in RNA space. But the framework is generic. You can assume, since in the spatial world, we already know what are the nearest neighbors, we don't have to integrate data set. We already can approximate using the expected or taking average across the nearest cells or the spots. In the paper, we show actually we can use this framework to improve the spatial expression of the data sets. Similarly, you can assume taking this as an example for sharing information between epigenomics and the transcriptomic space. And that's what we wanted to work further. Also, I would like to mention this Bayesian algorithm was not one-off tool. It relies on many subcomponents which were optimized in many different papers we have been working over the years. Especially, I would like to mention selective alignment, which is an alignment algorithm, previous version of Elmin, Minnow, which is a simulator, Swish, which is a differential expression tool, which relies on bootstrapping of Elmin's estimates. At the end, I would like to conclude by thanking all my lab members, especially from Combined Lab, Michael Love's lab, and also my current lab, which is Satija Lab, and NSF and IH for funding our lab. Thank you. I'm open to questions.